definition of this is the statisticians use sample statistics to estimate population parameters. For example, sample means are used to estimate population means, sample proportions to estimate population proportions. So why the estimation? Estimation means what? So, a point estimate of population parameter is a single value of statistic. For example, the sample mean x is a point estimate of the population mean mu. Similarly, sample proportions is a point estimate of the population proportion. So, internal estimate. An interval estimate is defined by two numbers between which a population parameter is said to be lie. For example, a less than x less than b is an interval to estimate of the population parameter mu. It indicates that the population mean is greater than a but less than b. So suggest x1, x2, etc. xn mu sigma square then mu equal to expectation of x, that is integration x d of x mu, where sigma square equal to x square d of x minus mu square, where mu hat equal to mu f, that is integration x into d of x, that is 1 by n summation xi. So here sigma square is x square d of x minus x bar square, that is mu square, that is 1 by n summation xn minus x bar whole square. This is also a non-parametric parameter as the normally assumption has not been ex- explicitly used. That is x1, x2, etc. xn be a random sample. So let x1 be a random sample from the following box. This is P1, P2, etc. Pk, where interested in parameters P1, P2, Pk and Q1, Q2, Q of P1, P2, Pk. Example, dividing the job in the population by 5 categories, interested in P5. So consider problem of estimating the upper estimated quantile QB and defined by 1 minus f of q, where probability of x greater than q, where u less than a less than 1 by n. The classical parameters approach consists of the following steps. Assume the parametric approximation f equal to f naught, where f naught is a generic cumulative distribution of a classic probability distribution model. Example, normal exponential gamma Weibel. This assumption is usually checked through some GOS test, that is chi-square Kolmogorov-Smirnov Kramer, von Moises, Anderson, Jelling. Compute and estimate theta on the basis of the sample statistics x1, x2, etc., xn, where estimate q as qa equal to f inverse 1 minus a. So this approach can lead to very bad estimation when d less than 1 by n. So Wilson in 1994. So here, since cumulative distributions issued from various parametric models can have the same shapes approximately, and the observation range minimum 1 less than x less and maximum xi, while exhibiting very different tail behaviors. To avoid such difficulties, specific methods for estimating extreme quantiles. So here, the most of these make use assume that F belongs to the domain of attractions. The extreme value distributions, Gumbel, Weibel, and Free Church. So these make can be grouped into two main families. Pic- Pican's approximation methods, only observations x exceeding a high threshold u are considered. The probability distribution of these excesses, the probability distribution of x minus u conditionally on x greater than u can be approximated for u sufficiently large by the generalized Pareto distribution. This method, called hereafter excess method, has infinitely been presented by Pickens. It has been studied by Smith. So, among others, two variants of the tail and the quadratic tail methods. So, they often provide better estimations than the original access method. Methods of estimation of parameters. So there are several methods of estimating the parameters available in the literature, namely method of movements, method of maximum likelihood estimation, the method of minimum chi-square, and method of generating best asymptotic normal estimates. So among these, we perfect the method of maximum likelihood because maximum likelihood estimate are asymptotically unbiased. They are consistent. So the very important one is in the maximum likelihood estimation in statistical theory. And please go through these points and how to get the maximum, like how to define the maximum likelihood estimation and the properties. Okay. So here they are asymptotically normal, they are asymptotically efficient, they are functions of the sufficient statistics. They are invariant in the sense that if g is a constant function of theta, the unknown parameter, the MLE of g theta is equal to g. Evaluated the MLE of theta. And MLE can be interpreted as the mode of the parameter density of theta, where uniform prior density is assumed theta. Maximum likelihood estimated iterative scheme. Here we provide iterative scheme for obtaining maximum likelihood estimates that can be performed by the use of a hand calculator. Of course, those can be programmed on computer if you so desired. Suppose we observe the series of pairs of observations x u, where x equal to log dose for a certain parameters, the standard one. Since we will dealing with arithmetic mean of response, it is not unreasonable to assume the normal distributions for the response. Let u be as normal u sigma squared, thus the log likelihood function capital L equal to constant p minus summation ui minus u whole square by 2 sigma square, where the constant involves sigma and not other parameters, and the summation is overall the response. Notice that ui involve the unknown parameters alpha and beta u equal to k, where alpha plus beta xi, where at p is the parameters denote the MLE values of alpha and beta respectively. There is dou L by dou A equal to 0, dou L by dou beta equal to 0, where A, B are some sample approximations to A and B obtained by some method of then A2 equal to A1 plus delta A1, B2 equal to small b1 plus delta B1, where delta A1, delta B1 are given by dou L by dou alpha 1 plus delta A1, dou square L by dou alpha 1 square plus delta B1, dou square L dou alpha 1 dou beta 2 equal to 0. Dou L by dou beta 1 plus delta A1 plus dou square L dou alpha 1 dou beta 1 plus delta B1 dou square L dou alpha 1 dou beta 1 dou beta square equal to 0. Suffix 1 alpha and beta indicates that the partial derivatives are evaluated at alpha and b. Considered dou L by dou alpha equal to sigma square sigma summation ui minus u into dou u by dou a. An interval estimation of the MLE only can replaces the second partial derivative of L by their expected value, since the expected values can be tabulated, and by the strong laws of large numbers. The second derivative converges to their expected values, where u equal to u after differentiating 5.2 we obtain expectation of dou square L by dou alpha square equal to minus sigma square summation dou u by dou alpha whole square, where expectation of dou square L by dou alpha dou beta equal to minus sigma square summation dou u by dou alpha into dou u by dou beta, where expectation of dou square L by dou beta square equal to minus sigma square summation dou u by dou beta whole square. We assume that all the responses are arranged in terms of the elements of a single vector. This is not all x values are distant, and there are may several u values at the same dose level x, where dou u by dou y equal to h of y, where y equal to alpha plus beta x, and define the weight function w equal to uh, h of y whole square. Thus, one can derive by 5.23 as expectation of dou square L by dou alpha square equal to minus sigma square summation w dy by d alpha whole square minus sigma square summation w. Expectation of dou square L by dou alpha dou beta minus sigma square summation w xi, and expectation of dou square L by dou beta square minus sigma square double square x square. Corresponding to the initial estimate a, b, expected value of response parameter capital Y is given by y equal to a plus bx, where dou a delta a into summation wx equal to summation w minus ui by h of y, where delta a summation wxi plus delta b summation wxi square equal to summation wxi into u minus u by h of y. So now added summation wy equal to summation wa plus bx to both sides of the first equation of wxy to both sides of second equation. If the response capital Y is defined capital Y equal to small y plus u minus u into h of y inverse. So here small a summation w plus b summation wxi equal to summation wy. So where delta a summation w plus these are the um, uh, equations of least square estimates. B summation w x square equal to summation w x y. So now equation 5, the linear straight equation is y2 equal to a2 plus b2x can be obtained by the weight linear regression equation y on x. Now x bar equal to summation wxi by summation w, where y bar equal to summation w uh, y capital Y by summation w, where summation equal to summation w x square minus summation w x whole square by summation w, where summation equal to summation w x y minus summation w x into summation w y by summation w, where b small b equal to summation by xy by summation xs, where a2 equal to y bar 1 equal to b2 x1. Now the process can be iterated with a2 b2 replacing a1 b1. The iteration can be stopped when approximation does not differ appreciably from the immediately preceding one. So notice that the working response capital Y given by does not conclude with the h equal to u unless h of u equal to u. However, we always expectation of y equal to h of u, u equal to expectation of u, where variance of capital Y equal to sigma square by summation w, where w equal to limited w, where and j denoting the stage of iteration. So here summation w and summation xs will be close to the values obtained at the last iteration. The unknown sigma square will be replaced by the estimates s square based on the residual sum of squares, namely the sum of deviations of the individual values of capital U about the estimates regression equation. In summary, it is desirable to have a table values of the um, mathematic transformation that used. A table of values of y equal to h of u is required by a table of the weighted coefficients given by equation. The minimum working response is y equal to y minus u into h of y inverse, where capital A equal to 1 by h of y. As function y can be made, notice that the big suffixes indicating the stage of iteration are dropped equations. First, we read the empirical response corresponding to each observed capital U, namely y equal to h inverse u, from the table, and then plot against x a straight line a I drawn by I through those points. Some allowance for unequal weights can be made when presenting the line. Expected response y r read from the line which corresponding with the observed x values. From the second table, the weighing coefficients for each y read and corresponding working response is formed as capital Y equal to small y UA. Then the weighted regression capital Y on X calculated now with the new set of values for expected response. A second iteration can be performed. The process of iteration is stopped when the pair of coefficients A, B differ very little from the pair A, B, and the last set is regarded as the evaluate values alpha and beta. The process of iteration coverage rapidly and it should not take more than two or three iterations. So the initial values AB are chosen judiciously. For quantitative response of the original observation are homoscedastic or um, statistically transformation for has been performed, then y equal to u, h of y equal to 1, where capital W equal to 1, capital Y equal to u. And the interactive process described earlier simplifies to calculating the unweighted. The unweighted linear regression and clearly one cycle suffixes in several practice situations. So this is the table where gives dose level Z 1 unit and 2 units. Next, let x equal to log Z, where small y equal to h inverse u log u. The points x, y are, these are the points. One can plot the points, draw a rough line. Let us the line be one of the two points. So next, y bar denotes the average of five y values corresponding to x equal to 0. The approximate equation of the line is this. So regression coefficient b equal to, this is the value. Next, small a equal to y bar minus b to x bar. So small b3 equal to this, b4 equal to this. So the MLE alpha and beta are approximately. So this is the equation. So here some of the questions: discuss asymptotic distribution. What is exact distribution in the statistics? Discussion Feller's theorem. Regression approach to estimate dot response and relationships. So asymptotic distribution of the ratio estimators Feller's theorem. Regression approach to estimate dose. So next one is the table. This is normal Atrel test and B1, B10, 14 point Atrel text. So this S square, this RR. So here regression approach to estimate the response or relationships. So here we will get the regression value small r z by h of x equal to h of or z. So here some questions. Asymptotic distributions of ratio estimators, Feller's theorem. So these are the one theorem that is, please go through this theorem. It's a very simple and you practice it will. Asymptotic normality. So here, parametric estimation. Although the R&M procedure is completely non-parametric since it does not assume any form for mx or h of yx in several cases, especially is quantal response situation where h of y by x known manually except for the values of a real parameter small y. We can representation such way that gamma is the parameter to be estimated, where expectation of y of x equal to m of x, x variance of y of x equal to variance of x, and these functions satisfy the conditions imposed earlier. Since y determines the model theta is a function of alpha and gamma, we further assume that there is one corresponding theta as h of x. Now the problem is choosing small a in order to minimize the asymptotic normal variance or the estimate of gamma. So one can easily show that n power of 1 by 2 h of x minus y is asymptotically normal with mean 0 where variance is h of theta whole square sigma square c squared by 2ac minus 1. So now the example. efficient. Now, RM procedure under SACS assumptions with an observation sum of theta known, where in case discuss the UP and down rule, quantal response is function up and down rule. So, Dawson and Moore have proposed a method of estimating LD. So, weight is simple than the RM procedure. In the latter, the dose levels are random and cannot specify them ahead of time. The Dixon Moore procedure chooses a series of equally spaced dose level Y. The response is observed only at themselves. We take the first observation at the initial guess of ID, where D is dose level. If the response is zero, the next trial is made to the immediately preceding dose level. If the positive response is coded as 1 and no response is denoted by 0, then the data any look like the one in figure is 0. Let the response be 1. Then we go dose 1 and response is 0. Then we go dose 0 and the response is 1. Then we go dose 1 at which the response is 0. Then go to 0 dose at which the response is 0. Then go to dose 1, the suppose the response is 1. Then go to 0, response is 0. Then dose 1, the response is 0. Then go to dose 2, the response is 1, etc. And this is a method not appropriate for estimating dose levels other than LD because of the assumption that the normal or the logistic distribution will be violated in the extreme tails. So if y denotes an estimate LD, the tests are made it where y equal to y naught plus or minus ID, where D is an estimate standard deviation, capital Y is the level of the initial test, where P equal to phi of y minus u by sigma, then M equal to summation M, and assuming n less than or equal to, we can write the asymptotic likelihood function. So here, so these are the equations, please go through. And here, this is the some questions in examinations. They will ask, explain the optimum sampling strategy, discuss the polycotomous quantal response, what are Blum's coordinators, discuss parametric estimation, what is a Monro process, what is the concept of UP and down rule and quantal rule, and what are the concepts of polycotomous quantal response. Explain briefly the concept of Robbins-Moore, Penrose, and Robbins-Moore procedure. Explain the concept of Bloom's conditions, asymptotic normally, linear approximation in the estimation of points on the quantal response function. Explain briefly the UP and down rule in the estimation of points on the quantal response function. Explain the concept of modified UP and down method. And here explain the concept of gene and genotype. And here Mendel had a Weinberg glass estimation of allele frequency, multiple allele. And here concept of gene and genotypes. The main objective of genus is importance of statistical genetics. Know the concept of genes and genotypes, and the allele frequency and relative frequencies of allele and genotype. And here statistical genetics is the use of statistical methods to find the genetics. The basic complex diseases, statistical genetics is a scientific field concerned with the development of statistical methods for drawing inferences from genetic data. The term is most commonly used in the context of human genetics. Statistic genetics tends to collaborate closely with genetics, molecular biologists, clinicians, and bioinformatics. Statistical genetics, a type of computational biology, statistical genetics generally involves developing theory or methodology to support research in one of these relation areas. One is population genetics, one is genetic epidemiology, one is genetics. So, population genetic is the study evolutionary process affecting genetic variation between organisms. And here, the genetic epidemiology is study of the effect of game genes disease. The quantitative genetics is the study of the effect of genes on normal phenotypes. The concept of genes. The term gene was introduced by Danish botanist, plant psychologist, and genetical William. So these genes are often called the blueprint of our life because they tell about the each of the cells. It may build a muscle, make bone, carry nerve signals, and so on. How do gene or alternate all this? The make protein, in fact, each gene is really just a recipe for making a certain protein. So here the characteristics of gene are as follows. Gene are the functional unit of heredity, variation, mutation, and evolution. They are situated in chromosomes. Every gene occupies a fixed position in a chromosome. They are arranged in a single linear order in a chromosome as beads on a string. In a broad sense, the term genotype refers to the genetic makeup of an organism. In other words, it describes an organ's complete set of genes. In a more narrow sense, the term can be used to refer the alleles, variant from a gene, that are carried out by term can be used to refer the alleles or variant forms of gene that are carried out by an organism. Humans are diploid organism, which means that they have two alleles at each genetic position. In sweet pea plants, the gene for color of a flower has two alleles. One allele codes for purple flowers and is represented by the uppercase letter F, whereas the second code for sweet plants could therefore feature these possible genotype as a locus, where FF, small ff of flowers. A particular genotype is described as hom- homogeneous of it features two identical allele and heterogeneous. If the two alleles are differ, the process of determining a genotype is called genotyping. Population genetics is very quantitative. A description of the genetic structure of a population is seldom simply a list of genotype, both rather uses relative frequencies of genotypes. Genotypes frequencies lead to allele frequencies. With quantification comes a certain degree of abstraction. For example, to introduce the notation, genotype, and allele frequencies, we will not refer to particular sample, but rather to locus that we all simply call the locus. Initially, we will assume that the locus has two alleles called A1 and so segregating in the population, these two alleles are different state. There will be three genotypes, that is A1, A2, A1. So genotype A1, A11, A12, A21. Relative frequency X11, X12, X12. So here X11 plus X12 plus X22 equal to 1. So allele frequencies where P equal to X11 plus 1 by 2 X12. Q equal to minus P equal to X22 plus 1 by 2 X12. Where small p equal to X11 plus X12 plus 1 into 1 by 2 plus X12 into 0. Where here p equal to xii plus 1 by 2 summation xi plus 1 by 2 summation xjj. Where here suppose 25 out of 750 students to red heads, what is the frequency of red heads? If random student is chosen, what is the probability that our method red head? That is the percentage of frequency of red heads to 25 by 740 into 100, 10 by 3. That is 25 by 750 is 1 by 30. So here, if the genotype AA and AA have frequencies 0.5, 0.25, 0.25 respectively, where small p equal to 1 by 2 x1, 1 small p equal to frequency AA, 1 by 2 frequency AA, where small p equal to 0.5, 1 by 2 into 0.25, 0.625, where check your progress, genotype AA and have frequencies 0.25, 0.25 and 0.50 respectively, then P equal to frequency of AQ equal to frequency of. So here some exercise, what is genotype, what is difference between gene and genotype, explain the concept of gene and genotype, what are the characteristics of genes, statistical genetics, concept of genetics, concept of genotype, how to relative frequencies of allele genotype. And please note down these exercise summary and check your progress. So today I am finishing this and please go through this. If you have any doubts and ask me. And today and please go through this Once again, I am repeating. And tomorrow I'll going to explain Mendel's Hardy Weinberg laws. So this I will explain later. So next, this is the topics. We have next estimation, allele frequency, multiple alleles. Next, approach to equilibrium for linked gene. Next, natural selection, mutation, and gene credit. Next, equilibrium will both natural selections and mutation operative. So this is the model paper and please go through this. And these topics are very small. So this is brief introduction I'm giving you. Est- Brief introduction I'm giving you Mendel's Hardy Weinberg laws. The main objective of this is Mendel laws. Describe the scientific reasons for the success of Mendel's experimental work. Identify the differences in Mendel laws. How to apply the probability rules in Mendel inheritance. So here Hardy Weinberg law assumptions. So introduction is he did deduced that gene common pairs and are inherent has distinct units, one from each parent. Model tracked the segregation of parental genes and their appearance in the offspring as dominant or recessive traits. So here the first law of Mendel is the law of segregation that describes the separation of the two copies of each hereditary factor or gene during the formation of uh, gametes. Each gene exists in two copies called the alleles in a diploid genome. So each allele comes from each parent during the formation of genetics. The allele pair separate from each other in such a way that each gamete recovers one allele from the pair. Therefore, the offspring acquires one copy from each parent. During the fusion of gametes, it acquires two alleles from each parental gamete. Alleles can be either homogeneous or heterogeneous. One allele in the heterogeneous pair is dominant, while the other is recursive. The expression of the dominant allele it produces the phenotype is called complete dominance. Shown in the figure, the Punnett square describing Mendel's first law by a monohybrid cross. The phenotype is ratio between the offspring in Mendel's first law. So this is the Mendel's second law is Mendel is the law of independent assortment. This law describes how alleles of different genes independently segregate from each other during the formation of gametes. However, it is applicable only when two or more factors are inherited together. According to Mendel-Holtz's second law, the transmission of different alleles or different genes into the genetics is not affected by each other. Mendel describes the second law with a dihybrid cross. The phenotype ratio of a dihybrid cross shows the behavior of the character short nail, long tail, brown coat, and white coat is a hybrid cross. So here the law of dominant an organism with alternate form of gene will express the form that is dominant. The main difference between Mendel's first and second laws is that Mendel's first law of segregation, the separation of allele pairs from each other during gene information and their pairing duration fertilization, whereas Mendel's second law of independent ascendant, so describes how alleles are different. Genes independently segregate from during formation gametes. So this is the class I explained, and please read each and every topic. Thank you, VRM, OU Thank you, Vijay Kumar.